Hello, this video is to help you learn to use the codon charts that you will need to be able to do for your SOL. So there are two kinds of charts. There's either the circular one or the square one, and I don't know which one you're going to see on your SOL, so we're going to go through both of them. So in the process of making a protein, transcription takes place in the nucleus where the DNA at a gene is read to make the mRNA. Notice that there are no T's in the mRNA. The A matches to a U. And then that mRNA, once it's made in the nucleus, will travel out to the ribosome that's in the cytoplasm, and the ribosome will read the mRNA as part of translation. Now, when you have your mRNA, the first thing you probably want to do is divide it up into its codons, which is every set of 3. So I'm going to separate these out into the 3 codons. Now, so let's do our first codon here, CUA. So when you're dealing with the circular chart, you always start in the center. So I'm going to get a line here, and here's my C. So the first letter here, C, means I'm at this C right here. So what's next? Well, it says CU. So I need to go from this C to this U, and I'm going to change the color of that line here so you can see it a little better. All right, so I'm going from C to U. Well, the last one in our codon is A. So I'm going to go from C to U and then U to A. And so now I'm going to A, which is right here. And notice that takes me to LEU. All right, so I'm going to go over here. LEU is the amino acid that goes from C to U to A. So that's how you read the circular codon chart. If we have GAU as our next, so now we're going to start in G. And at G, our next letter is A. So now I'm going to go from G to A. All right, so that's our A. And then I'm going to go from A to U, which is right here. So that for that third one, G to A, and then A, look at the letters attached to the A, find the one that you need, and we're going to end up with ASP. ASP. Notice if we had had GAG, we would have ended up at a totally different amino acid. All right, so next we have UAA. So let's start in our U section. So here's our U. And we're going to go from U to A. All right, so here's UA. And then we're going to go from A to another A, U to A to A. And notice we end up with that little box there, which according to our code means a stop. And stop means stop. If the ribosome got to UAA, it would stop making the protein. It would not add any more amino acids after that. So stop indicates the end of the protein. Whether that's intentional or there was a mutation, stop means stop. All right, the last one we have here is AUG. So again, we're going to start in the A section. So now we're in A. And we need to go from A to U. So here's our A to U. Okay, A to U. And then our last one is G. So I'm going to come over just a little bit here. And it's just this single one right here, AUG, and it says MET. Now, that is the name of the amino acid. A lot of times you see this little green arrow that says start, and that means that in most eukaryotes, AUG or MET is the first amino acid in most of the proteins. Not all, but most, So AUG is a little bit special because MET is the start protein or start amino acid. Okay, so that's how you read the circle. That's how you read the circle. Start in the center and then just work your way out based on what is attached there. All right, so let's try the this square one. So notice it's labeled first, second, and third. So here we have our mRNA. We're going to divide it up into codons, into every 3, so that we have 3. And now we're going to have our codons here. So CAU. So this time we want to find the first letter. Here's the C. And for that one, I'm going to come over here and start at C. And I know that the whole row is C, right? This whole row is C. My second letter is A. So here at the top, I find the A. And I'm going to place my line there. And you can see that in that column, C and A meet at this box. So let me highlight that box here. Give me just a second to highlight the box. Okay, we're going to make this clear. Oops, make it clear. And then I'm going to make it really stand out. So I know where I'm focused. I am where C and A intersect, which is this box right here. Now I need to look at my last letter, U. Well, if you look over here on the right, U is the first letter. So that U means that the first one that is listed in that row is the one that I want. And so that means that HIS is my amino acid because U is the very first one here. All right, let's try the next one, GUA. So G, here's my G column. Let me get a new line, row. And so I know I'm in this row. Why not make it a different color? So we'll make it green. All right, so GU. All right, so let me copy that. And this time I need U. So I need the U column. All right, and you could just do this with your fingers. I'm doing it here on the computer. All right, so G to U, GU. I know I'm in this box right here. And then A is the third one down. So if I were to point to it, the A one here is pointing directly to this VAL. VAL. Now, you'll also notice that it doesn't matter where I am in that box, they all say VAL, which kind of helps when you have mutations. You can have the same amino acid even with a small mutation. All right, now let's do UGA. So our first letter is U. So we're going to go over here and let me change my color to blue. All right, so now we're in the U row. All right, I'm in the U row. I need G this time as my second letter. So let me get my G, and we're going to find where those two intersect. So U and G intersect in this box right here. I need A, and notice here our A is the 1, 2, third one down. A is a stop. So STOP, stop is stop. All right, last one, AUA. So let's get our first row here. We are now in the A row. Let me change this to even a different color. All right, so we're in the A row. Now we need U. Okay, so let's go to where A and U meet. Oops. Right, so A and U. So, I mean, we're reusing U, but this time we're going to look at a different row. So A and U intersect right here in this box, right? This box right here, and I need A, which is the 1, 2, third one down. So this time it's ISO. ISO. So A is the third one down. Let me move that up a little so you can see it. All right. So that's how you read either the circle chart or the square chart. Circles start in the center and radiate out. Square chart, look at the first and second to see what box they meet in, and then you will just figure out of those 4 that are inside that box, which is the last one that you need. All right, I hope that helps.